MCQs of topics that we have covered till now. Welcome back to BioBoost everyone. Today we will cover all the MCQs related to the organelles that I have covered in my videos: nucleus, mitochondria, ER, ribosomes, lysosomes, cell cycle, cell junctions and cell signaling. These will be very helpful for part B of CSIR-NET. So first MCQ, which is which of the following is structure is responsible for selective transport between nucleus and cytoplasm. Here in this diagram you can see nucleus is there, nucleolus is there inside it. The outer covering is the nuclear envelope. If we see the transverse section, then here you can see a nuclear pore complex is present. Here central pore is present. So cytoplasm is outside of it. So on the nuclear envelope it is present. So the selective transport of nutrients and everything is take place between nucleus and cytoplasm is the nuclear pore complex. Rest, we see that nuclear lamina is just a fibrous network of proteins, and remaining nucleolus and chromatin. Nucleolus, you can see it is present inside of the nucleus. So it is not the passage between nucleus and cytoplasm. So it is responsible for synthesis of rRNA, ribosomal RNA. Ribosomal subunits are assembled inside nucleolus. So these are the roles and function of nucleolus. Chromatin is just a complex of DNA and proteins. It is not responsible for any kind of selective transport. So, so the structure which is responsible for selective transport is nuclear pore complex. So our option will be B. Here you can see these are large protein complexes and they regulate entry and exit of RNA. Next MCQ is mitochondria related, which mitochondrial component is directly involved in ATP synthesis. So here in this diagram you can see the outer and inner layer of mitochondria. On inner layer you can see ATP is present and ETC, electron transport chain, is taking place. Outside outer membrane has porins and transporters. So we in question you have to see directly word, you have to emphasize that directly which one of the mitochondrial component is involved. So inner membrane is involved where oxidative phosphorylation takes place, where electron transport chain takes place, and it also has an enzyme called as ATP synthase enzyme. So directly involvement of ATP synthesis takes place on inner membrane of mitochondria. Matrix is not present because it has just enzymes. So outer membrane is, you can see, because of porins and transporters it is permeable to small molecules, while intermembrane space that accumulates protons, right? The space present between inner and outer membrane is intermembrane space. Here protons get accumulated, while matrix has enzymes for Krebs cycle. So our correct answer will be inner membrane where ATP synthase enzyme is present. Also, cristae aids in ATP synthesis. It is not a direct involvement, it is an indirect involvement where cristae increase surface for ATP production. Next MCQ is smooth endoplasmic reticulum is mainly involved in. So we have to see the differences between smooth and rough. Smooth has no surface bound ribosomes. On smooth endoplasmic reticulum, there are no ribosomes present. So our first option will be protein synthesis will be incorrect here. Next, RER is attachment of ribosomes are there. Next difference is formed of tubules and vesicles, while RER is made up of cisternae and few tubules. Next, SCR is connected with plasma lemma, while RER is connected with nuclear envelope. In SCR, synthesis of glycogen and lipids takes place, while in RER, protein synthesis takes place because ribosomes are there. Next, in SCR, detoxification takes place. So enzymes for detoxification are present in SCR, while in RER, no such enzymes are present for detoxification. In SCR, they are developed from RER, while rough endoplasmic reticulum is formed from nuclear envelope because it is in continuation attachment with it. So if we see the options, no protein synthesis, no ribosome, So here lipid synthesis takes place. Next MCQ is free ribosomes in cytoplasm synthesize proteins that are mainly destined for. If we see a normal cell, then here RER is present, on it ribosomes are present. Those which are free get transported to cytosol and rest are gone to lysosomes. So free ribosomes and attached or bound ribosomes, if we see the difference, then free ribosomes, they float freely in cytoplasm. So ribosomes which are free in cytosol and attached are attached to endoplasmic reticulum. Next, free ribosomes synthesize proteins within the cell. Whatever proteins are made from these, they synthesize proteins within the cell, while bound ribosomes secrete, means they release secretory ribosomes. So if we see the option, then free ribosomes are for cytosol and nucleus. Next is lysosomes are called suicidal bags of the cell because we have to focus. We are not asking what is the function of lysosomes. It is asked specifically why they are called the suicide bags. So we have to take the related option to it. We know the functions involves waste digestion, recycling, defense, apoptosis, cell death, and plasma membrane repair. These are the functions of lysosomes. But in option, we have asked why they are called the suicidal bags. So we have to focus on it, and that's why we focus on the C option, which is cause self digestion of cell, because it ruptures, membrane ruptures, and the enzyme digest by autolysis. Next is cristae are present in mitochondria too. We need to focus on what is the functional part of cristae because they are present to store ATP. No, we first focus on the functions of cristae. So it is responsible for ATP production, not storage. So our option A will be incorrect. It is the primary site for oxidative phosphorylation we covered on above. Second MCQ. Now, second is function is increased surface area, means because of extensive foldings, it has an increased surface area. So our option B will be correct here. Increased surface area for respiration. Contain mitochondria, it is not its functional feature. Transport proteins, no, that is related to transporters on the outer membrane. So option B will be correct. It is for increased surface area for respiration. If we see the diagram of mitochondria, the inner membrane, and these are the infoldings of cristae, and it is the outer membrane. So outer, inner, and cristae. So option B will be correct, where increased surface area for ATC and other oxidative phosphorylation reaction. Next is nucleus. The main function of nucleolus is, we are asked for nucleolus and not nucleus. So here you need to focus. If we see a plain cell, then here cytoplasm, nucleus, and inside of it nucleolus is present, and a cytoplasmic material is present, and inside the nucleus chromatin is present. So if we see, chromatin is responsible for DNA replication, so here nucleus is part of that. Protein synthesis take place in ER and lipid synthesis in SER. So our correct option will be ribosome formation, where we know ribosomal RNA is produced and assembled in the cell of ribosome. Next is of cell junctions. Which cell junction prevents leakage between adjacent epithelial cells? Here you can see in diagram the major cell junctions and cell matrix junction. We need to focus that desmosomes are responsible to link cells via intermediate filaments, while gap junctions, as the name suggests, there is a gap, so there is a communication. So these create channels for direct passage of ions. It does not prevent any kind of leakage. And hemidesmosomes, these anchor cells to outside extracellular matrix. So you need to focus on the word prevention of leakage and also between epithelial cells. So option B, which is tight junctions, it prevents seal any kind of leakage, and it is also present in epithelial cells, right? So if we see the function of tight junctions, then we know that these are responsible to seal tightly between cells, and these are also abundant in epithelial tissues, right? So our in epithelial cells wise, our option tight junctions will be correct. Next is for cell signaling. Here if I make a diagram of cell signaling, a GPCR receptor is there, ligand is there, and a ligand is coming, then G protein is getting activated, and a secondary messenger is there, which is CAMP here, and it activates GTP into pKa, and then cellular response takes place. So here if we see the role of secondary messenger here, then it is to amplify. Here if we see the role of secondary messenger is to amplify the initial signal by activating downstream enzymes, right? So one ligand activates many secondary messengers and it causes signal amplification. So our option B will be correct here, to amplify extracellular signals. Next MCQ is mutation that prevents calcium release from sarcoplasmic reticulum. If we have studied the sliding filament theory, then it suggests that when a thin filament, that is the actin filament, it get attached to troponin heads with tropomyosin, then in the presence of calcium ions only, the filament is contracted. So muscle contraction takes place only in presence of calcium ions which are released from sarcoplasmic reticulum. So here our option second is correct, where muscle contraction option is there, and as sarcoplasmic reticulum stores calcium ions, calcium release initiate the actin myosin interaction here.